Hello there! My name is Mog, yes, that Mog. And today I'm going to be talking about chemistry, more specifically organic chemistry. Now, I'm a chemist and a practicing scientist. I've done research in a chemistry lab for the last about almost 2 years or so. Yeah, that's what I want to say, about 2 years or so. It's been a while. Point of the matter is that I am a practicing scientist, and I love science. I love it so much, it's awesome. But let's be honest here, organic chemistry sucks to learn. Yeah, it's a kind of class that has a reputation that very much precedes it. And it sucks that it sucks to learn. It sucks to suck, because organic chemistry is awesome. We have so many advancements as a society, because organic chemistry has progressed so rapidly over the past couple of decades. New synthetic techniques and advancements have brought us things like better pharmaceuticals, and better methods of making polymers for alternatives for fabrics for clothing. It's really great. And not to mention the fact that working in organics is just so satisfying, getting to make new compounds and figure out why molecules behave the way that they do. Here's an example as to why organic chemistry is so important and so awesome. Aromatic compounds. They're basically a type of molecule that's extra stable because they have alternating double bonds and single bonds. They're constantly used as scaffolding molecules in drugs because they're so stable. They help to form very stable backbones for drugs to do their business. It's really cool. You should give it a look-see. But let's be honest here. Organic chemistry really sucks to learn. And there's a lot of reasons why. And to help me cope with how much it sucks, I'm going to be playing this game. So let's get right into it, shall we? So the very first reason why organic chemistry sucks to learn is honestly the classmates. And more specifically, the dreaded pre-meds. Okay, look. I get it. First of all, I want to preface this by saying that hashtag not all pre-meds. Believe me, I know that there are good pre-meds out there who are smart, dedicated to their careers, and very much good people and good students. But the thing about pre-meds is that a huge amount of them are just total mouth breathers. They're people who don't care about their classes, and they're people who don't really care about medicine, really. They're kind of the type of people who, to put it bluntly, think that they can cheat their way through medical school the same way that they're cheating through undergrad. Now you see, when you're a chemistry major, you take a lot of different kinds of classes, ranging from calculus, to physics, to biology, to proper chemistry. And that also means that you're going to be taking classes with people who are not chemistry majors. In fact, you're more than likely going to have a very hard time finding other people who actually want to major in chemistry, even in your real chemistry courses. And that's because chemistry is a very broad and diverse field that touches along other types of disciplines. A lot of people who take organic chemistry have to take it because it's required for other kinds of degrees, especially things like biology, biomedical sciences, certain engineering courses, and physics. Now, obviously, there are a lot of really smart and dedicated pre-med students out there, but a huge amount of them are total mouth breathers. So many of them are always complaining about how much they hate all of their classes and how none of this is important for med school, quote-unquote. And that's especially the case when it comes to organic chemistry specifically. Here's a short anecdote. One time, I was waiting for an exam outside my testing center at my school, and I was talking to this one girl, or more rather, I was doing some last-minute review for my organic exam, and this one girl decided to just pass the time by making small talk. I don't know why, but she just did. And I asked her what her major is, and she says biomedical sciences, and that she wants to go to medical school. Nothing wrong with that. And when I tell her what my major is, she gives me this very shocked and surprised look, and she just goes, "What? Chemistry? How on earth could you actually like this stuff?" And she just goes on and on about how much she hates organic chemistry, and not just that, but all of her chemistry classes, actually, and how none of it is that important for being a doctor. Now look, I understand. A doctor who's practicing medicine, say in like an emergency room floor, is not going to need to know most of organic chemistry. I highly doubt that a practicing surgeon is going to need to know that, is going to need to know that potassium permanganate is a good reagent for syn dihydroxylation in the middle of surgery. I understand, okay? That's just not going to happen. But the thing is, organic chemistry is so incredibly important to understanding biochemistry, which is extremely important for medicine in all regards. You see, organic chemistry is the study of relatively small organic molecules. Organic just means that they're made up of carbon and hydrogen atoms primarily, with some other stuff on top of them. We also study a lot about how to make them and why they behave the way that they do. Biochemistry, on the other hand, is a little bit different. It's essentially the study of biopolymers, which in other words are large macromolecules that are organic in nature, and just so happen to be, you know, the main molecules of life themselves. They're the primary molecules that organisms create, which is why it's called biochemistry. I feel like a lot of pre-meds going into it go into biochemistry with the anticipation that it's just going to be biology 3, and then they start crying in the first week when they have to start learning about thermodynamics, which I find hilarious. Back to the main topic, however, things like protein folding, hydrophobicity, E2 reactions, nucleophilic attacks, and polymer synthesis, pH scale, pH versus PI versus pKa, all of these things are incredibly important to know in biochemistry, and you're not going to— oh shit, I didn't mean to— ugh. And you're not going to understand those without the background of organic. All of those things are topics that you learn in organic chemistry very intimately, and you're just going to be lost in the context when you go to biochem. So yeah, organic is definitely very important to know for medicine. If you still don't believe me, there's a couple more examples I can get into, like factor VII and proteases. In fact, serine proteases in particular are a very good example of organic chemistry at work in a biological setting, especially when it comes to things like the catalytic triad of serine proteases. Hello there, Future Mog here. So in the video, I mentioned something called the catalytic triad when I was talking about why organic chemistry is so important to medicine, and specifically understanding biochemistry. Now you might be wondering, what the hell is a catalytic triad? Well, you know those protein things that you've heard about before in food and stuff? So this right here is aspartic acid. It does not actually participate in the reaction very much, no, instead it mostly just plays a support role. You see this dotted line right here? Well, that's what's called a hydrogen bond. It's not a real covalent bond, like, you know, this right here, or this right here. No, instead, a hydrogen bond is essentially like a magnet. This oxygen is very negatively charged, and this hydrogen is very positively charged. Now, oftentimes in biology, we'll want to cut up proteins to do a lot of different things with them. And the way we chop proteins up is with a specific other kind of protein called a protease. In fact, the specific bond that we break in amino acids with proteases is called the scissor bond, quite literally because we are chopping it up with scissors. We call it the catalytic triad because it involves 3 different amino acids specifically in order for it to work. These are serine, aspartic acid, and cysteine. Now there's a specific mechanism that involves all 3 of these, which is often used in what are called serine proteases, named so because they all use serine specifically. Other proteases exist out there where aspartic acid and histidine aren't involved, but serine is by far the most common, and is sometimes listed as the most important. I'd argue that serine is more important for reasons that we'll discuss as we view the mechanism, but that's neither here nor there. Now you might be wondering why exactly we're going to be using amino acids to chop up other amino acids. Well, quite frankly, it's because we're using a protein to cut another protein. It just makes sense that we're using the same components to chop up other components. I promise it'll make more sense once we get into the mechanism itself. So let's just make some space here and get started on drawing. So first of all, amino acids all look like this. H3, there. So this is the basic form of an amino acid. This is just what they generally look like. On the left side here, or sorry, right side here, we have what's called the carboxylic acid terminal. This entire thing right here is called a carboxylic acid, and the protein will just kind of continue on from this side. On the left side, we have what's called the amino terminus. This NH3 right here is called an amine group, and the molecule will also continue from the left side here. When you continue an amino acid, it's going to go on from the oxygen to the nitrogen bond. So what that looks like is sort of like this. And the rest of the molecule just kind of goes on this way for a while. This specific bond right here is what we want to break in order to chop up the protein into the left half and the right half. And this is what's called the scissor bond that I was talking about before. So say that you have an amino acid that looks kind of like this. This here. Okay. Say that you have an amino acid that looks something like this. It's a bit awkwardly drawn, but whatever. And we want to cut down this specific bond. This is the scissor bond, and we're going to separate the protein into two halves. How do we do that exactly? Well, this is where the catalytic triad finally comes into play, and we're going to have to do a bit of setup first. Alright, so here's the basic setup. Over here we have what's called aspartic acid. By the way, I'm only going to be drawing the R groups. These are the functional groups that are important. I'm not going to draw the entire molecules because that's going to take too much time. Opposite electric charges attract each other, and just like magnets, these are going to get closer to each other and pull the electric charge away from the rest of the histidine molecule, over to the left. That means that this nitrogen right here is going to be more susceptible for what we need to do in just a bit. We also have the serine right here, which is about to do what's called a nucleophilic attack onto this part of the molecule of interest. But we're getting ahead of ourselves. So first things first, the lone pair on this nitrogen is going to attack this hydrogen, and then the bond from this hydrogen is going to go onto this oxygen, making it more negative, and from there, it will attack this carbon specifically. Now the thing about carbon is that its valence electrons look like this. It has 4 available electrons, meaning that it can make a total of 4 bonds at one moment. You'll notice that our carbon right here already has 4 bonds going for it. 1, 2, 3, and 4. Because we already have 4 bonds, we have to push one of the electron bonds here onto this oxygen, making it extra negative. And then from there, we're going to go on to our next step of the mechanism. By the way, because the aspartic acid doesn't really play much of a role in the rest of the mechanism, I'm just going to leave it as a little squiggly line right here, just to let you know that it exists, but it's not very important for anything else going forward. So I'm just going to stop talking about it. So the general setup is that you'll notice that the nitrogen right here, just stole the hydrogen that originally came from this part of the molecule right here. That gives it an extra positive charge. And even though nitrogen is better at holding positive charges than other molecules, it's still pretty unstable. But more importantly is this part right here. Like I said before, one of the electric bonds from this double bond between the carbon and oxygen jumped onto the oxygen molecule, making it extra negative. That's why I wrote down this little minus sign right here. The oxygen doesn't want to be this negative, so it pushes the electrons back onto this bond right here to form another double bond. But we still run into the same problem as before, where this carbon cannot hold more than 4 bonds. So because of that, it's going to lose one of these 3 other bonds right here, and the bond that it's going to choose to break is this specific one, the one that's connecting it to the rest of the molecule, because remember, this R1 means the entire left half of the protein. So that bond is going to be broken, and then jump onto the nitrogen. From here, these electrons on the nitrogen are going to try and bond to something else, and what they're going to choose is this specific hydrogen right here, which will alleviate the excess and positive charge onto the nitrogen, because the bonds here are going to jump back onto the nitrogen and then neutralize it. and that'll lead us to the third part of the mechanism. So the good news is that we are currently halfway through the mechanism, and congratulations, we already did it! Yep, our molecule is officially cleaved in two. This entire thing right here is the right half of the protein. But there's one problem. So you might have heard that enzymes are proteins that are known as catalysts. A catalyst is just any kind of molecule that will make a chemical reaction happen faster. And that's what these three bad boys did. They chopped up the molecule in a very particular way that made it go a lot faster than it would have otherwise. But the thing about catalysts is that they don't truly take part of the reaction. Not quite. A catalyst is defined as something that will speed up the reaction and always be present in the final part of the reaction. That is to say, we will always end up with what we started with. The catalyst will not be consumed. And right now that's not the case. We still don't quite have what we started with. The serine is covalently bonded to the rest of the protein, and we need to get it off. So how exactly do we get serine off of the rest of our protein? Well, this is where water comes into play. So we're going to have a water molecule, which comes in, and what's going to happen next is something called water activation, which is just a fancy way of saying an acid-base reaction. So our histidine right here is going to act as a base, and the water is going to act as an acid, whereby these electrons right here are going to nab up this hydrogen. And just like before, the bond here has to go somewhere else, and it will go onto the oxygen, giving it an excess charge of electrons. So we know how this works by now. The electric charge has to go somewhere off of this oxygen, and it's going to go to the most positive place it can find. And in this case, it's going to be this carbon right here, for the same reasons as before. The electric charge is going to be pulled and stretched in every which way, and this carbon is going to have a partial positive charge. So it'll drag onto here, and I should probably remove a couple arrows. So these electrons will move here, and like before, bada bing, bada boom, and we can move on to our next step. Okay, a lot is happening, but basically what is going on here is that we have the hydrogen that came from the water now attached to this nitrogen of the histidine. It acted as a base, and the water acted as an acid. In turn, the remaining OH itself from the water now bonded to the rest of our protein, and the protein itself is now attached to 3 different oxygens. It is attached to the carboxylic oxygen that it was originally supposed to have, it's still attached to our serine, and it's now attached to the remainder of this water. So how exactly are we supposed to actually get rid of all of this? Again, we're supposed to start off with what we originally started with. Well, second verse, same as the first. These electrons are going to push back onto this carbonyl bond right here and reform that double bond from before. But we don't have an amino group like we did before, so which bond of these other 3 do you think is going to break? The answer is the serine, where the molecule is going to go here, and then oh, well, actually, now that I think about it, I kind of drew this in a weird way, but basically, the bond is going to break, and the electrons are going to go onto this oxygen of the serine in green. And from there, they're going to attack this hydrogen right here on the histidine. So then we will get our final product of this. And there we go. This is what we have as our final product. Just make this really awkward box around it. So yeah, this is what we started with. We have the aspartic acid, which didn't really play much of a role, just kind of hydrogen bonding with this N right here. We have our histidine, which looks exactly the same as before. We have our serine, which picked up the hydrogen that originally came from water, and we have the thing that we care most about, the cut in half protein. This is the right half, by the way, that we originally started with. It looks the same as any end of a protein should, with a C terminus, with this hydroxy group that came from the water, and the double bonded oxygen, which was always here. Here's the entire mechanism, by the way, in case you wanted to see it in full. It's a bit hard to follow, but I hope I explained it pretty well. Like I said before, this is incredibly important to learn in biochemistry to understand how proteins actually carry out their functions. More importantly than that, however, it's a very common mechanism that's used in a lot of diseases, specifically their pathology, or in other words, how exactly a disease infects a human being. When you get infected by HIV, you might have heard that what it does is inserts its own genetic code into your cells. Well, the way it does that is by using protease enzymes. It quite literally uses this exact mechanism in order to insert its genes into yours to produce more HIV cells. One of the most common treatments for HIV are a type of retroviral drug called protease inhibitors. Their job is to stop this entire mechanism you see here. If that doesn't explain why organic chemistry is super important to understand for medicine, I honestly don't know what does. But your classmates are only one part of the problem. Another part is just the fact that classes just suck. They really suck. Organic chemistry, like I said before, is a class that a lot of different people have to take. Chemists, pre-meds, biology majors, physicists, certain engineers like chemical engineers, material scientists even. And probably more that I can't even think of. As a result of so many different kinds of people having to take organic chemistry, the classes end up being absolutely massive. It also doesn't help the fact that a lot of the majors that do require organic chemistry as a prerequisite or main requisite also just so happen to be very popular majors. In fact, I believe in 2022, some of the most popular majors included biology and health sciences, which had 131,000 and 268,000 graduates respectively in that year. So classes end up being absolutely massive, with hundreds of students. Me personally, my section of Organic Chemistry 1 at my school was taught by an adjunct professor with 230 students, with a maximum of 250 for that section. The missing 20, by the way, is because of those 20 people who just dropped the course midway through the semester. So yeah, a lot of classes are typically massive and stuffed with way too many students, and universities don't always have faculty or staff to teach a lot of the sections for it. A lot of schools just tend to spread themselves out very thin on resources, so to speak. Teachers very often don't teach this class very well for a variety of reasons. Mostly for a lack of professional interest or poor communication skills, skimming over foundational content that's important to understand other things, quote-unquote because it was covered in Chemistry 1. Side note, I absolutely hate it when professors don't cover important foundational topics because, oh, it was just covered in high school or something. Ugh, I hate that. More often than not, these teachers tend to be adjunct faculty, which if you don't know what adjuncting is, I'll link you to a video that explains it better than I could. Oh shit, oh god, oh boy, oh. Now, that's not to say anything about adjunct faculty. I don't think that they're bad at their jobs per se. I mean, a lot of the adjunct faculty that I've had the displeasure of being taught by were bad, but in all seriousness, plenty of them are good at their jobs. They are teachers who like teaching. Now, being a tenured faculty or an adjunct does not indicate the quality of pedagogy. Rather, the need for contracting people to teach several sections of a huge classroom that's already being stretched thin for resources. Oh god. Uh-oh. Okay, I really need to pay attention to this game. Oh god. Oh god. Now, like I said, tenured versus adjunct doesn't really indicate the quality of teaching you're going to get in an organic chemistry class. What I'm trying to say, though, is that the need of contracting so many different people just to teach a massive class that's already being stretched thin for resources, it's a symptom of the problem, so to speak. We all learn in different ways, but being in a huge classroom just makes it more difficult on everyone to learn such a difficult topic. This is part of the reason why there's 1.5 million different organic chemistry explanation videos on YouTube. Shout out to the organic chemistry tutor, my beloved. He's my real professor. Because of the massive amount of students, disinterested professors, and course materials that are usually not made in-house and are usually farmed out to third parties, like Pearson and Third parties like Pearson, Wiley, McGraw-Hill are often used to assign homework to students. They all have varying degrees of quality. Screw Pearson. Pearson may suck and McGraw-Hill might be subpar, but hey, at least Wiley isn't that bad. But the sheer volume of homework assignments in this class is just so insane. I distinctly remember my very first week of Organic Chemistry 1. My professor had assigned us one homework assignment that was 50 questions long, right? It was due about 11 days after the class had first begun. The second homework assignment, which was also 50 questions long, was due 12 days after the class had begun. How the hell are we supposed to do all that in such a short time while still learning the material? This sort of spaghetti scheduling just makes it worse for everyone involved. It's a very difficult course that's not taught very well, that's taught by very disinterested people to very disinterested people. But if you're a fellow STEM major, you'll know that that's not all. No, far from it. Because organic chemistry is also a science course, this means that there's also going to be a lab component. Like I said in the beginning, I'm a scientist, and I love working in the lab. It's really fun, and I could talk about my work all the time, for hours on end. But lab assignments, it's just like being in hell, but from the discomfort of your own local college campus. The organic chemistry lab just sucks so much. Now, again, I have to put a disclaimer, this is just from my experience alone. I can't tell you how your experience will go if you're about to major in chemistry or something that requires organic. But the way it was for me is that it was basically just a series of chemical separation and analytical techniques. Things like thin layer chromatography, distillation, rotovapping, and short one-step syntheses. Now, the actual lab procedures themselves are not very hard. There's just a bunch of prep work that they want done for your notebooks, like a reaction scheme, the reagents and hazards, SDS info, drawings, theoretical yields. It's just a lot, and that's all for the pre-lab stuff. This isn't even talking about the post-lab workups that they make you do. Now, the pre-meds come back here with a vengeance when they start complaining about how they never learned how to calculate theoretical and actual yield, which is just a load of crap. I know for a damn fact that these people learned how to calculate theoretical and actual yields in Chemistry 1 and 2. That's one of the very first things that they teach you in that class. The theoretical yield stuff in particular was a really big sticking point in the chemistry lab I was part in. It was so dire that the GTA literally complained 3 weeks in and did a mini lecture at the beginning of lab on how to calculate your yield, because so many people just kept not doing it right. Not to flex, but multiple people just started copying my own homework because they didn't know how to calculate the yields themselves. And what can I say? Actual chemistry major in the building kinda has its perks. Now, other than the labs, there is one more thing that's really, really big sticking point for organic chemistry as a whole, as a thing to learn. And that is the dreaded, the big boss of chemistry itself, the ACS exam. Now, for those of you who don't know, the ACS, or the American Chemical Society, is an organization in America that does a lot of promotion of chemistry as a field. They don't do research themselves, but they do publish and fund other people's research. They also have a bunch of things like scholarships for chemistry majors and people in related fields, and they also do a bunch of fun programs, and they even have their own local chapters at a bunch of colleges. They have a bunch of neat things going on for them. Something that isn't very neat, however, is that damn ACS exam. The ACS is an accreditation institution for most large school universities. Chances are, if you've ever heard of a university, they most likely have their chemistry program accredited by the ACS. And part of ACS accreditation includes forcing students to take standardized exams that are made by the ACS themselves. Wait, hold on. What upgrades does this thing have? I think I'll just go for this and, this. Yeah, that'll work. Now, the ACS exam is really hard. It's really, really hard. And it comes back with a bigger vengeance in Organic Chemistry 2, where it's also the final exam there. Problem is, for Organic 2, the ACS exam also makes you recall information from Organic 1, as well as general and analytical chemistry. That is a huge amount of things to make students prepare for just one class. And the thing about the ACS exam, right, is that it's a standardized test, okay? It's also not made by your professor, it's made by the ACS themselves, and there's a huge amount of privacy and security around the test itself and its questions. In fact, if you look at the front page of the exam, if you ever take it, the very front page is an instruction sheet to the professor telling them absolutely do not look at the questions. So yeah, your professor doesn't know what's going to be on the exam. It could very well be things that you didn't even get taught in your own organic chemistry class. That's how hard of an exam this is. It's also close to 100 questions long, and chances are you might not even have enough time to do it. The average exam length is, what, an hour and a half, 2 hours? I was thankful enough to have extra time on mine, to get 3 hours, but I used up every last minute of that, and I was going insane by the end of it. It's just so much unnecessary stress for a class that I don't personally think needs to be that hard. Organic chemistry, yeah, it's a broad field with a lot of information to learn, but over the course of 2 semesters... Oh, hey, I won. Nice. Woohoo. But over the course of 2 semesters, I really don't think they need to shove that much information and make it that obtuse and difficult to understand. I'll just do free play now. Let's see how far I can get. Comanche defense. Well, actually, wait, hold on. I'll get Comanche defense, yeah. And then put down a second super monkey, why not? There we go, yeah, sure, there, I guess. So yeah, the class itself being really hard and needlessly frustrating, coupled with a lot of students who are very demoralizing to be around, quite frankly, it just really, really sucks. And the worst part is, it doesn't need to be this way. Because, believe it or not, organic chemistry can actually be so much fun. Wait, no, please, no, don't leave me, I can explain! Now, I've worked in organic research labs before, and I currently do synthetic work in a biochem lab, and it's so much fun. It really is a lot of fun. I love working in this lab. Getting to work on new compounds is super fun and rewarding and so cool, especially if we talk about different synthetic techniques. And if you think that, you know, making chemicals in a lab isn't that cool because you yourself have done a chemistry lab at school and absolutely hated it because it was a lot of work and not fun at all and super confusing, I get it, okay? I understand. But seriously, I think that you should give organic synthesis another shot. Maybe not doing it yourself, but definitely look up the Cubane series by Nile Red and Extractions in Fire. It's a really great series that is a lot of fun to watch. And the excitement that those guys get just making Cubane, as well as a bunch of other stuff, that's how I feel when making chemicals in a lab myself. It's just so rewarding to finally get a unique molecule that you've been synthesizing for days on end. Me personally, the stuff that I worked on had to do with photocatalysis in one of my previous labs, and that was just so much fun, because it's a way of inducing chemical reactions in such a unique manner by... Oh wait, hold on, what upgrade should I get with this thing? What does this do? Okay, yeah, bottom path sounds better. But yeah, photocatalysis is really fun because it's such a unique way of inducing reactions. Basically, the TLDR of it is that you shine a ultraviolet lamp at a reaction, and the energy of the light that you shine on your chemicals causes the electrons to jump into a higher energy state, making them more susceptible to react with other electrons. And that's just one example of how organic, but chemistry in general, is just a field that's all about finding creative ways to solve problems and make cool things happen. I feel like if organic chemistry wasn't a weeder class, it would really inspire a lot of people to, you know, pursue a career in chemistry and science in general. And organic chemistry is awesome because it can encompass so many different disciplines and subfields. Oh god, okay. Chemistry utilizes a huge, wide variety of different things just to get things done. We use lasers, giant magnets, blazing heat, freezing cold, explodey stuff, burny stuff, like sodium borohydride, which is really fun to work if you lift work with if you live in the southern United States where there's tons of humidity. Yeah, if you couldn't tell, I have a penchant for working with all the dangerous stuff. It's weirdly fun for me. I don't know why. Oh, no, 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 please don't die, please don't die. Oh god, I'm gonna, oh, wait. Aw, damn it. Oh well, that sucks. Well, at least I made it to round 95. Totally like I don't, always die here in this game anyway. Back to the topic at hand, though, I really do think that if more people were exposed to organic chemistry, or chemistry in general, in a more lighthearted, less cutthroat way, a lot of people would find it really great and might want to even pursue more careers in chemistry. I feel like creative and critical thinking really does wonders for people who get more exposed to just how to do that. Because in the physical sciences like chemistry and physics, you're not doing a lot of memorization, honestly. It's not like biology where you just have to memorize a bunch of names and what they do, and that's about it. No. In chemistry, you need to do a bunch of critical thinking to understand what's really going on and make predictions about a system. Critical thinking is also important to just fix problems in science. You need to be creative as a scientist. One example with me, actually, is back to that photocatalyst experiment. For what we were doing, we were using toluene as a donor for what's called a hydrogen atom transfer, which is exactly what you think it is. It's basically just transferring a hydrogen atom to another atom, to another molecule. My bad. Ugh, cannot speak tonight. We were having some trouble reading the analytical results of our experiment, and to clear up the fuzz and make the overall thing a little bit more efficient and clear up our data, we used deuterated toluene to prevent the HATs, the hydrogen atom transfers, and just get a better idea of what was going on. Deuterated toluene, by the way, just means that the hydrogen atoms are replaced with deuterium, which is hydrogen 2, basically. It can't be detected by an NMR machine, which is what we use to analyze molecules in organic chemistry. But yeah, this is just one way of where you have to think really critically and creatively in chemistry and science in general, just about how to perfect the conditions you need to get the product you want and how to maximize your yields. In the end, though, I think organic chemistry as a course just really sucks all the fun out of it, and it makes it seem so much less enjoyable than what it really is. You know, maybe if more people got better exposure to what research as a scientist is like in a real academic research lab, and not one of those lab assignments, then a lot more people might actually like it. But who's to say? After all, I'm just a talking moogle who really likes to go on and on about chemistry, data, science, and academics.